Hello, I'm Kari Lavikka, a PhD student from the University of Helsinki. I'll talk about Jellyfish, which is a new visualization design we developed within our recent study on tumor evolution in ovarian high-grade serous carcinoma. We are also developing a software package that automatizes the visualization process. The paper was published in Cancer Cell a few months ago. We studied clonal heterogeneity in high-grade serous carcinoma and found 3 evolutionary states, which are also characterized by distinct pathways. However, in this talk, I'll focus on Jellyfish. But first, a few words about high-grade serous carcinoma and tumor evolution. Treatment of high-grade serous carcinoma often comprises neoadjuvant chemotherapy and several debulking surgeries, enabling acquisition of samples at multiple time points during the treatment. High-grade serous carcinoma originates from fallopian tubes and metastasizes to different organs in the abdominopelvic cavity. Thus, we also get samples from multiple anatomical sites. In the study, we had a discovery cohort of 55 patients and validated the results with another 93 patients. Carcinomas are initiated by genomic aberrations. As the cancer cells proliferate asexually, they inherit parent cells mutations but also gain new unique mutations. The evolution forms a tree, which is called phylogeny. When we get tumor samples, they comprise mixtures of subclones, which are cells representing different branches of the phylogeny. In the study, we were particularly interested in heterogeneity. In other words, what kinds of mixtures of cells the tumor has and what kinds of evolutionary relationships they have. We used computational methods to infer subclones and the tumor phylogenies. First, we used PyClone6 to cluster somatic mutation allele frequencies, and once we had the clusters, we used CloneEvol to infer the clonal ordering, in other words, the phylogeny, and compute the final clonal compositions. CloneEvol also produces a visualization, but it wasn't satisfactory, because an obvious problem is that there's a dendrogram for phylogeny and separate bell plots for each sample. Because of the lack of integrations, getting the big picture from this visualization is very difficult. But how to visualize this? There are some existing solutions such as Fishplot and ECCB, but we didn't find them satisfactory. But we came up with a set of requirements. Multiple anatomical sites should be supported, and also multiple time points should be supported. But the most important one is the third one. Integrates tumor's phylogeny and the clonal composition of each sample. So an integrated approach. Then there are more like minimal redundancy and maximal readability, and also efficient use of space. Here are some early design ideas from my colleague Dr. Jaana Oikkonen. And for instance, this one actually looks a bit like the jellyfish you are eventually gonna see. But she also had experiments with other designs like a forest of phylogenies, where the areas of the nodes depict the subclonal frequencies. But this becomes quite difficult to reason about when the number of samples and subclones grows large. So I'll now talk about the visualization design that we developed iteratively. The Jellyfish you see are largely drawn manually by me using Affinity Designer, and there are a total of 55 jellyfish in the paper's supplement. But we are developing a tool that aims to fully automate the process. Anyway, its physical sample is represented by a rectangle. The right edge shows the observed subclone frequencies in the sample. If the subclone was first observed in this sample, it's visualized using a bell that depicts logistic growth. The samples are arranged into columns that represent different time points. Now we see two relapse samples. The right one was acquired later. To make it easier to follow the subclones, we connect them by tentacles. Let's add two diagnostic samples that were acquired from different anatomical sites in the same operation, so ascites and peritoneum. But how do we know where the subclones came from? Well, we don't, but we make an assumption. If there's an earlier sample from the same anatomical site, that is the origin. And we have two ascites samples here. Otherwise, they are inherited from an earlier, possibly hypothetical sample. To illustrate the phylogeny of the subclones into two diagnostic samples, we add a hypothetical inferred root sample, which is shown as the bell of the jellyfish. Finally, we add some driver mutations that were first observed in specific subclones. Here we have three jellyfish that represent very different evolutionary trajectories. In the first one, there's little change in time and space in the subclonal frequencies of the samples. In the second case, we have samples only from one time point, but there's plenty of divergence. So many of these samples have unique subclones. We also use inferred samples here, which are shown as bells. The idea is to highlight the clades in the phylogeny and also reduce the number of tentacles in the visualization. The third example shows an interesting case where in the diagnostic samples we see quite uniform subclonal compositions, but after the chemotherapy these subclones disappeared and have been replaced by totally new subclones. Really interesting case. Alright, there are multiple phases in drawing the plots, applying to both manual and automatic method. I'll explain each one briefly. Redundancy wastes space and makes it more difficult to find a good layout. But which of these are redundant? Well, those with similar clonal compositions and the same time point and anatomical site. So not all three are redundant, but these two primary omentum sample. They could be combined. But how to find the similar samples? We can use relative entropy or other divergence and apply hierarchical clustering to find samples that can be combined. Then extracting inferred samples. Well, in brief, if the descendant of a specific subclone occurs solely in a specific group of samples, and the samples contain no other subclones, we can extract an inferred sample. maybe a bit tricky. I'm not going into details now, but anyway, here it's easy to see that P over R2, P over R3, P per 1, P ohm, they share a common ancestor, this subclone. But also both P over R2 and P over R3, they have subclones with a common ancestor. Okay, computing layout for the samples. The exact algorithm for computing the layout is still in progress, but we have identified several requirements. First, similar samples should be placed close to each other, for instance, based on divergence. Also, crossings of the tentacles should be minimized because crossings make it really difficult to read the visualization. And also tentacle length should be minimized and the plot size. The current implementation is quite unsophisticated, but we are looking at constraint solvers or maybe simulated annealing, but that happens in future. Okay, drawing the samples is largely solved. So there are two main requirements. The proportions should be shown clearly, and we are using stacked layout for that. Also, phylogenetic relationships should be shown clearly. And here, instead of stacking, we spread this. And to actually generate this layout, we use nested smaller step functions. And we use the same algorithm for both inferred and real samples. And this is nicely parameterized. Then drawing the tentacles. Well, an efficient algorithm is still an open question. Here's an example of a non-trivial routing problem. I have drawn this manually, so I had a chance to, make these tentacles go between the samples nicely and find a proper layout. But doing this automatically, it's difficult. So maybe constraint solvers could be used, or maybe printed circuit board autorouters could be used. Well, not used, but they might offer some ideas. And then the final phase is to add driver annotations. I came up with some design guidelines, like entering the arrows in 90 degrees angle, and they should approach from the left. But how to place these labels and arrows? In the Vega project, there was a paper about label placements, and they used occupancy bitmaps. That's an interesting approach, and maybe we could also use that. So, here are some jellyfish produced by the tool my colleague and I are working on. While the current version already outputs jellyfish, there's still quite a bit of work in getting all the details right. Once we reach the sufficient level of functionality and correct output, we will publish this as an open source tool. To conclude, we successfully used the visualization design in our recent paper. However, semi-manual drawing is both time-consuming and error-prone. I spent quite some time drawing these 55 jellyfish, and I'm not gonna do that anymore. So we are developing a tool that will enable automatic visualization of jellyfish. And we will apply this tool to much larger cohorts, having hundreds of patients. And there are some example use cases like discovering differences between primary and metastasis sites and evaluating the evolution of tumors during treatment. I have now been talking about cancer, but maybe this tool could also be used with other organisms that have asexually reproducing populations, like bacterial cultures or something. But yeah, that's all. Thank you.